Welcome to Think Tech on OC16, Hawaii's weekly newscast on things that matter to tech and to Hawaii. I'm Jay Fidel.
And I'm Duke Oishi.
In our show tonight, we'll cover how Dr. John Paul Bingham is unlocking the mysteries of indigenous snails at UH Manoa.
We'll talk with Dr. Bingham and meet some of his snails and research assistants. We'll see one of his assistants milk a local snail for toxic venom.
When we met with Dr. Bingham beforehand to discuss his work, here's what we learned.
The College of Tropical Agriculture and Human Resources, CTAR, our founding college at the University of Hawaii, is focused on agricultural problems as well as engineering problems which are going to benefit Hawaii, whether it be biodiesel or the development of new plants or new agricultural materials. My specific interest is in biopharmaceuticals, but also in pesticide agents. And a lot of the work that we do is focused on some of the problems that affect Hawaii specifically, being in a tropical environment, in regards of pests, trying to control these certain organisms from either damaging our agricultural economy and also having some cases, some of these pests having effects on human health.
Mm-hmm. What's your credentials?
My credentials, I did an undergraduate degree in biological chemistry and majored in clinical biochemistry as a graduate student. And that was at the University, Griffith University in Queensland, Australia. I then did my PhD at the University of Queensland in the Centre for Drug Design and Development, where that stimulated a lot of my concepts in regards to my area of expertise in these particular marine snails, which you'll see. So I then went to UCSF, University of California, San Francisco, learned more techniques there, then to Yale Medical School, another couple of years there, and finally to Northern New York as a research scientist, very close to the St. Lawrence River, very cold up there. And then finally got the opportunity to come to Hawaii as a tenure-track assistant professor in the Department of Molecular Bioscience and Bioengineering. And this is a very unique department within an agricultural college that we bring in the hard benchtop science, we bring in the agricultural interests and problems and implications to the research that we do in the test tube. And we try to build these transitional elements into the environment to allow people to get benefit. Our stakeholders, our agriculturalists, our local people who want to have information regarding certain substances or use on their plants, we can try and provide that type of information.
So what's your laboratory like? What's your team like?
My team's a very interesting group of students from all different types of backgrounds, nationalities, but we're all drawn together for a focus on peptide chemistry. Now, we are basically synthetic chemists. We do a lot of stuff in test tubes and wear white lab coats. But we also have a unique branch in our research area where we incorporate some of the natural products or compounds that we find in Hawaii, which are unique, and in our environment. And that we try then bring those compounds in to research and development. So the students usually have a very wide interest from marine biology to synthetic chemistry to ecology to animal sciences to plant sciences. So we're kind of a mix match, a cooking pot of ideas, and that's a very good lab to bring together, because then when we have a problem, we don't all see it from the same perspective. We will have heated discussions in some cases. But the idea is that we're trying to move the research material through to get results, to get cures or to find new compounds or to do something to solve a problem that Hawaii may be facing.
Yeah, with that kind of diversity, you have a greater possibility of creative thinking, out-of-the-box thinking.
That's right. And we've got one particular group of students who really sit down and do a think tank and say, okay, here's the problem, how are we going to analyze this? But basically, we've all been trained in the scientific method, developing experiments, hypotheses, and then trying to develop then finally the correct pathway we should lead our investigation or research. And it's, as I said, it's a very unique environment to be in charge of. You know, the ideas and stimulation that I get personally as the professor, as the principal investigator for my students, is quite amazing.
And you can see that.
Oh, yeah.
In my conversations with you, I've always noticed that. Someday I'm going to be in your class, you know.
Okay, that's okay.
So now, you know, so people shouldn't worry and wonder what we have here on the table. These are what? Snails, and you're working on snails.
Yes. These are a group of marine gastropods we find here in Hawaii. Now, The Hawaiians know about them, and if you go into the South Pacific, all the locals know about them, but not very many people from the mainland know about them. These are known as cone shells, and they are actually a family of poisonous snails. They belong to the family called Toxoglossa, which in Greek is poisonous tongue. And these particular snails we're working with to develop new pesticides and new pain medications, which not just affect Hawaii, but have implications worldwide. So these groups of snails are found in the tropical waters around Hawaii. We snorkel, scuba dive them. And previous people and other researchers around the world have demonstrated their pharmaceutical properties already. And we think Hawaiian specimens are very unique, that they've been isolated through evolution, and that potentially they may house a few more compounds which we may not see elsewhere. And so we're doing some very interesting research on these.
Why don't you just tell us what they, the particular snails involved? And by the way, to clarify for our listeners, these are not live snails, and you can touch them and not worry, right? You can touch them and not worry.
So the snails themselves, there's about 600 species to the genus, so they're a very large family, and they're and when we talk about food pyramids with predators and hunters, they're right at one of the top of the apex, because they don't have very many predators, but they kill a lot of things. Some eat fish, and these particular species will usually sting with a hypodermic needle or a tongue, and they'll shoot at about 200 meters a second, so really fast, pretty fast going. And they can go through a 5-millimeter wetsuit, so you've got to be careful how you handle them.
And then they'll inject a toxin.
That's correct.
That could be fatal.
Could be fatal, particularly with the fish eaters, because in our body we have similar physiological events which also occur in fish. And just by chance, some of their toxins cross-react with our physiological effects and then cause respiratory suppression, cardiac arrest, but interestingly, dull the sensation of pain. So this is what some of the people started thinking about regarding how they could make analgesic drugs from these. So we also have species which also eat mollusks. So these guys, and usually have these white little tented pattern marking.
It's very aesthetic.
Oh yes, very pretty shell. Will eat other marine gastropods, including their fellow brothers and sisters in the family. They actually are carnivorous and cannibalistic. So when we put them in tanks, we've got to be very careful of sorting them out.
So the head of the snail comes out, and the snail has a mouth, and the snail eats its siblings.
Yes, so what it'll have, it has a little siphon or a nose, and it smells for where the prey is and moves towards the prey. Now, the old saying is being slow as a snail doesn't really apply to these guys. These guys move pretty fast.
Then we spent some time with Dr. Bingham and some of his research assistants at the College of Tropical Agriculture at UH Manoa.
My name's Clifford Kopono. I'm from Hilo, Hawaii. I'm a second-year graduate student in Dr. Bingham's labs, and I work on characterizing conopeptides and looking at their interactions on organisms such as fish, snails, and worms. A peptide is a small single-strand chain of amino acids. They're a small molecule pretty much that make up what proteins are, and that's what we study in the lab.
Hi, I'm Zan Halford. I'm a master's student for John Paul Bingham. I also use peptides. I have synthesized a typical toxin from the scorpion that's used in a lot of potassium channel studies. It's a tool to help create new drugs for symptoms such as arthritis, epilepsy, cardiac issues.
What he's actually doing, he's taking nature as a template, and he's actually chemically engineering in a test tube materials which we supply to a lot of other labs on the mainland and worldwide, because no one else in the world is making these or investing the technology that we do in making these compounds. We have people at Yale, at Stanford, at Harvard, at Brown, who use our compounds specifically for investigating their diseases or their particular applications in medicine. So it's very important for us to be able to produce this material chemically, because the only way you can actually produce this stuff, you can't use DNA, recombinant technology, or other processes. We have to use it chemically. And so he is actually building some new building blocks, which will then expand this field of making these toxins as research tools. Cliff, on the other hand, he's looking specifically at nature itself. That nature is providing a number of compounds which we can get from the snails. In this case, these are snail toxins, conotoxins or conopeptides. And he is needing to...
We also visited his special snail room. We met some of his snails and one research assistant who had the courage to milk toxic venom from those snails.
But our main focus here is we supply the venoms from the cone shells. So we keep the cone shells in the tanks and basically feed them fish and acquire their venom in a small little container like this. And I'll get Jeffrey, one of my master students, to show you how they do that and see the viewpoint of the snail as it attacks and deposits its venom.
My project has everything to do with Conus striatus and the variability of their venom. So when we get these snails into captivity, we see enormous variation in their venom from the time that we get them into captivity to various life stages that they go through to any number of other factors, and we really don't know what these factors are. I like to call it Pavlovian conovenomics, which is the study of conus peptides, conus venoms, and the response stimulus reaction that they have with their environment. So what we're doing is we're very carefully looking at the life histories of these animals. When do they lay eggs? When are they mating? When do they go off of feed? The various seasons, moon phases, just a few of the variables. And this is just scratching the surface in the beginning stages of a project of this nature. And at the same time, we're tracking their venoms to see what peptides are increasing in volume, which ones are decreasing, are any new ones showing up. And that way in the future, hopefully we can provide these guys with optimal conditions under which we can get, let's say we want protein or peptide X, more of it expressed. So we can say, well, we need to milk them during the full moon at such and such a temperature, and we'll get it. What we use to extract the venom from these is it's an Eppendorf, which is a standard vial you'll find in just about any chemistry lab. We've snipped off the top to it and taken a latex, a cut up condom membrane to go over the top of it. And we've just secured it in place with a rubber band, and this is what we use as a target and the collection vial for our cone snail. There's nobody on record in Hawaii as having died from cone snail stings. So that's a good sign. In fact, only 15 people worldwide, to my knowledge, between 15 and 30, they believe, have actually died directly from the cone snail sting. Pick a victim. This is probably a small fish for them. We've got a guide of just pictures of each of the snails that I use to identify them based on their unique markings that they have on their backs. Then I put the scales in there for just a little bit of scent. Entice the snails a little bit. So that gets the scent going around so the snails know it's dinner time. You'll have to kind of bear with me because they're snails, so they're not very fast. But once they get going, they will. You'll see a little bit of action here. You can see this guy on the left. He has a couple things sticking out here. He's got that's his rostrum, right there. This is his mouth sticking out here, and this little white stalk, I don't know if you can see it, is his eye. So what we're looking for is his proboscis, and you'll know it'll come out of his mouth, and it will be bright orange. And that's what he uses to subdue his prey. There, you can see just the end of it now. Now he can reach that thing just about the length of his shell if he has to. So what we do so let me see if I can do this. So I'll just cover up our membrane with the tail of the fish. See, sometimes they just get too friendly with the front glass here. There we go. There. Oh. And he just stung the fish. I missed the venom sample on that one. He shot the venom straight into the fish in that one. So what happened was he got a fleshier part of the fish than just the tail. And sometimes, because it's tough to see with the fish moving around and him moving around all over the place, he'll hit a fleshier part and it just doesn't go all the way through. So I want him to hit like the very thin membrane on the base of the tail, or at the end of the tail, and it'll go right through that membrane and into this one. So let's give it another try with this guy. He's looking a little bit excited here. I missed that milking, JP, for the first time in... No, I know, I'm slipping. There it is. So if you look very closely, you can see his mouth opening up, and there's a line attaching the Eppendorf to the snail. So I just go in there and snip the line.
No, yeah, I can see it now. It's sitting on one.
Is that it? That little tiny thing?
That little thing
Yeah.
Yep.
is the remnants of the harpoon.
It's roughly the size of an eyelash. After all that, I've got what I came for, and that is an Eppendorf with our radula in the top, and we can take that to our electron microscope for later analysis. And at the bottom, that's how much venom you get. So sometimes it's a little bit more, sometimes it's a little bit less. Just for reference, it's about 20 microliters right there. And that's enough to kill a fish, that's enough to kill a much larger animal, and in some cases, that's enough to kill a human.
The funny thing is you can see all the snails are all up and active now, because what happens, they smell the protein from the fish introduced and they all become highly excited.
Yeah, you can see the proboscis thing. Is that what
Yeah,
you
the siphon, and look at these ones climbing up the glass.
Dr. Bingham is a first-class biotech research investigator. His laboratory and research projects are a great asset to UH.
You never know where research like this can go, but it certainly seems that discoveries of global consequence could happen in Dr. Bingham's laboratory right here in Manoa.
We were very impressed with Dr. Bingham and will continue to follow his work. And now, here's our Think Tech calendar of events going forward.
This week, on August 25th, the Hawaii Venture Capital Association and ThinkTech will present a luncheon panel program at the Plaza Club called Newsmorphosis Redux, dealing with changes in our local news media since the closing of the Honolulu Advertiser and Star Bulletin a year ago.
Next month, on September 22nd, we'll present a luncheon panel program called Impact Investing and the New B Corporation, dealing with socially responsible investing and the new B Corporation form of business, which Hawaii has adopted to help make that possible.
On October 27th, we'll present a luncheon panel program called Update on Agriculture, featuring State Agriculture Director Russell Kokobun, to get us current on developments in diversified agriculture in Hawaii. To sign up for these programs, visit hvca.org or thinktechhawaii.com.
And now, here's Bill Spencer with this week's Spensation.
You know, in terms of, business and investing, September 22nd, we're going to have our impact investing program. And we're going to talk about B corporations. Can you tell our listeners a little about that?
Today, you really have to have a sense of a triple bottom line, meaning not only profits and productivity, but social responsibility. That has to be built into the business model. And this is being recognized globally, and certainly in the United States, by this concept of impact investing, which is investing in businesses that are having a social impact as well as a performance bottom line type of impact. There's a new form of organization out there that is called a B corporation. It's starting to take hold. In Hawaii this year, a law was passed that will allow that form of organization to exist. It's sort of a hybrid between a nonprofit, which is a social do-good entity, but it's fashioned in the for-profit modality. Why can't you do something good and be profitable? What's wrong with that? I don't think there is anything wrong with that. And so the B Corp fills that gap, and I think it would make more sense for investors. Investors don't just need tax breaks. They can invest in things that'll have a positive benefit.
No, we should learn about it. It may change our way of looking at investing, and it may change our economy, and it may change our startups too.
Well, like I say, I think startups have to have a social benefit component. You know, we've always talked about corporate responsibility, but that's when we're dogging the big profitable corporations to sort of give back to society. But they've already made their money. What about the guys who are starting up and want to do something that's beneficial? I think that has to be factored into the investment decision matrix. And it may take longer for companies like that to return a profit. They may require more money up front than your typical sort of .com advertising play. But the benefits can be much, much greater in the long run.
Yeah, there is investment in the community, and the community benefits, and as a member of the community, you benefit.
And, our community's global these days.
Yes,
so
it is.
We have to keep that in mind as well. Even big ideas can have a social benefit.
Welcome to the 21st century.
I'm there, baby.
Thanks a lot, Bill.
All right.
And here's this week's episode about the adventures of our co-working entrepreneurs, Richung Fujihira and Hassan Scott.
We've been able to sign on 2 new clients that we're going to get exclusive use of long-term use, 6-month blocks at a time, which makes it very exciting to do and make it very viable to work with. We're also continuing with the North Shore expansion and seeing to be able to really leverage the beauty and, I feel more importantly, the idea of doing business in the beauty and community space of North Shore. And so we're working closely with a landowner out there to be able to try it out. And so with this, leveraging this idea, I think we'll be able to break into a whole new market of co-working.
The target market is going to be very different for this North Shore space. Whereas the spaces in central or in the core of Honolulu are specifically for local people, with the North Shore space, we're looking for a certain type of local person, but also people coming from outside of Hawaii, and they want to have like a staycation where they can work and also enjoy the beauty of Hawaii, and that's really how we're reaching out towards them.
We're finding our spaces being leveraged and used in all new different ways. And last week we were talking about an independent producer that came down and looked around our space and actually wanted to use it to take shots for their new commercial that they're making for an insurance company. So they came down and actually used the corner of our space to take a shots for, but they also came back and used it to do film editing and got their work done. So people are starting to really see how they can use our space, which I think is the best way to be able to connect with any type of customer.
We also have a Lean Startup Circle Hawaii, and they're going to be based out of our group. So a lean startup is, if you're familiar with the lean method that Toyota has in their Toyota Way, lean manufacturing, it's taking the principles of doing everything and spending your money like it's your last money and various other things, and applying that to startups, modern day startups. And the Box Jelly is a lean startup, and we're going to be having other entrepreneurs that use this method coming in every other week and talking about the methodology and what they're doing with that. One more thing, the photographers are coming in from Hawaii Business Magazine. So we're going to be in Hawaii Business Magazine pretty soon, and we're pretty excited about that. It'll be my first kind of real photo shoot, so we're looking forward to that.
Yes.
And being a part of their co-working section. And we are Hawaii's first and only co-working space.
We've been looking to get on sponsorships that align with our ideas and what we're trying to accomplish with our business model. And then we're also attacking the print media. So we've been working around all the way around, and also still continuing on of doing the foot traffic of utilizing our youth still and going out and hitting the streets and doing face-to-face, still selling those prepaid certificate hours, and we're doing everything else to such. So the Box Jelly is a place to work,
A place to meet,
a place,
and a place to build.
Work the way you live.
Thank you very much.
We'll be right back to wrap up this week's edition of Think Tech. But first, we want to thank our underwriters.
Thanks to the Scheidler Family Foundation, which supports a number of educational, cultural, and charitable organizations, including ThinkTech.
Hawaiian Electric Company and its affiliates Miko on Maui and Helco on the Big Island are deeply committed to the communities they serve.
Galen Ho is a senior executive of BAE Systems, a global defense, security, and aerospace company, and CEO of CBI Polymers, a tech company in Hawaii.
Oceanit, another local tech company, is one of Hawaii's largest and most diversified science and engineering companies.
Don't forget ThinkTech. We're funded by gifts and grants from people like you. Please help us out by making a contribution on thinktechhawaii.com. It'll help us with all the things we do to raise public awareness about tech, energy diversification, and globalism in Hawaii. Thank you so much. Okay, Duke, that wraps up this week's edition of ThinkTech. Remember, you can watch ThinkTech on OC16 several times every week. Can't get enough of it, just like Duke does and all of our other hosts too. For additional times, check out oc16.tv.
You bet, Jay. For lots more Think Tech videos and for underwriting and sponsorship opportunities on Think Tech on OC16, visit thinktechhawaii.com. Be a sponsor and help us reach Hawaii.
I'm Jay Fidel. Thanks for joining us on ThinkTech.
And I'm Duke Oishi. Aloha, everyone.